this chapter officially belongs to the same with the previous one, so it is part of hormones, homeostasis, and reproduction. And it defines the approach to what we are going to learn, because, however, about reproduction, it is focused mostly on reproductive hormones, with some exceptions, but this is not the most complicated thing you have to know about reproduction, minus menstrual cycle, which is a bit more detailed. Let's go. Reproduction, as you probably remember, is producing similar creatures equipped with at least part of your DNA. In case of humans, which are organisms reproducing sexually, it means that each of parents will have about half of the genetic input to the offspring. But producing offspring is not only contributing with DNA. So the first thing we have to know is what actually reproductive systems are supposed to do in terms of reproduction. Some functions will be shared by both sexes. So both male and female reproductive system will participate in gamete production. Of course, male organism will produce male gametes and female will produce female ones, but still the kind of task is quite similar. The gamete must be produced physically, then stored till the proper moment, nourished, and transported to the place where it can perform its function. And it is in common for both sexes. Similarly, shared function is participation in fertilization. So both sexes will participate in delivering their own gametes to the site where they can interact and finally meet producing a zygote. But, as in many cases of sexual reproduction, for the female sex, there is one huge additional task, which is absolutely not something what males can do, and this is pregnancy. So it's the creation of a proper environment for embryo to develop, formation of placenta, which is the organ which connects the fetal organism to the maternal one, and finally labor, so helping the fetus to get out of the maternal organism. The set of tasks to be performed by the reproductive system will determine what is done by the reproductive system in terms of particular actions. And it will also determine what you have to know about the structure of each of reproductive systems. The male one seems to be quite complex, and actually it is, but luckily not all parts of a male reproductive system have to be known to you. So let's analyze which of these parts are things you should be able to recognize on the diagram, name properly, and finally briefly comment on its function. So this is a penis, which you obviously know, however I'm afraid not necessarily from proper sources, and you have to be able to name it. Here we've got testis or testicle, one testis, two testes. Please remember that as a term derivative from Latin, it will be specific in singular and plural. So here we've got testis. On testis there is a cap which is called epididymis. From epididymis the ductus will go out, which is called vas deferens or ductus deferens, and this is this tube going upwards. Then it turns around the urinary bladder, goes down, and from this moment on it will start participating in semen formation. So many glands will be appearing, by the way. Seminal vesicle is the first of them, it is here. Then we've got ejaculatory duct. So what was previously vas deferens now becomes ejaculatory duct, but similarly it is just a thin tube, and it goes further, crossing another gland, which is prostate gland, probably the one you know already by the name at least. Then all this tubing becomes urethra at the moment when it means the third of glands you have to know: bulbourethral gland or Cowper's gland, the smallest of them and the latest to be met, by the way. So once more, parts which you should be able to recognize and name is penis, testis, epididymis, vas deferens, seminal vesicle, prostate gland, Cowper's gland or bulbourethral gland, ejaculatory duct, and urethra. The rest of these details on this diagram may help to understand where exactly are all these parts placed, but you don't have to name them. And here, just in case, you got a list of parts, which is quite a good idea to recognize and name. Now let's focus for a moment on their function, because each of these things has something to do, and probably some of these functions are more obvious, while some are rather mysterious at this moment and very specific, even if you learn about it. So the most obvious are testes, which just produce sperm cells. Please don't confuse sperm with semen. Sperm in English is only the cell which is present in this secretion of the male reproductive tract, while the liquid mixture of sperm cells with various additions is called semen. Okay, so testes will produce sperm cells. Epididymis is a part which stores produced sperm cells and will assist their maturation. Vas deferens is just for transport of sperm cells. Ejaculatory duct will transport seminal fluid. So before it becomes ejaculatory duct, vas deferens contains mostly sperm cells. But from the moment when it becomes ejaculatory duct, this tubing will collect not only sperm cells but many liquids and dissolved substances, which finally compose semen, which is the proper environment for sperm cells not only to live in but also be delivered within to the female organism and support survival of sperm cells. So it is quite important. And now if it comes to glands, seminal vesicles will supply fluids, and the component is good idea to know is fructose, which provides energy to sperm cells, which are capable of moving on their own, but they need some energetic substrate for this movement, and this is for some reason fructose. Prostate gland will add many different fluids to semen, and you don't have to know composition of them. And if it comes to Cowper's gland, it produces mucus and alkaline solution to overcome acidity of the female reproductive tract. Finally, penis is this part which helps in putting sperm cells in the proper place, which is as deeply in the vagina as possible. Only if placed properly, sperm cells will have the highest chance of getting to the inside of female reproductive tract and finding the egg cell. That's enough about parts of male reproductive system and what they do. A female one, however, it has more things to do, looks much simpler, and there are less parts you should be able to recognize and name. Look and notice also the fact that this is a different projection. While the male reproductive tract was shown from the side because it is much easier to find all important parts there, if it comes to the female reproductive system, it is much easier to see everything and understand when you look from the front. What do we have here? 2 ovaries, 2 oviducts, and there are 3 different names you may find in literature, all of them correct. You may call them uterine tubes, you can call them fallopian tubes, or just oviducts. Whichever name you use, it's okay. Then there is uterus, and the innermost lining of uterus should be named and located separately because it is very important. It is called endometrium. Then there is cervix, which is the gate between the internal part of the reproductive system and this part which can be accessed from outside. And then vagina, this is the exact place where penis can get into to safely deliver sperm cells to the inside of female organism. So look, sperm cells finally can get in assisted way only as far as to the cervix, but they have to find oocyte which is more or less in the fallopian tube. So the rest of route across the cervix and across the whole uterus and across a part of fallopian tube, sperm cells have to go on their own. This is why they are able to swim. What about functions of all these parts? I assume that at least some of them are rather obvious. Ovaries will produce oocytes. Oviducts will transport oocytes, but there is also one additional function which many people seem to overlook. This is exactly where fertilization should take place. So not in uterus as many of you think, but in oviduct, what is actually earlier. Very, very soon after the oocyte is produced, it must be fertilized or it dies. So this is not like it is very likely that after being produced, the oocyte will meet a sperm cell and will successfully produce a zygote. If it comes to uterus, basically it will create a space in which fetus will develop. But if it comes to endometrium, so this innermost lining, this is a very interesting tissue because together with fetal cells it is capable of forming placenta, łożysko. So endometrium will be always discussed and described separately from the rest of uterus because its function is extremely important, actually crucial for pregnancy as a process, because only if the placenta is formed properly, a pregnancy is what it should be. So providing to a fetus from maternal organism. Cervix, as we said, is a gate which protects inside of a reproductive system from being accessed by something what is unwanted, but at the same time it of course should create a safe entrance for sperm cells if they arrive. And finally, vagina is a place where sperm cells can be placed without going outside to the external environment because it would be not safe for them. Humans performing so-called internal fertilization have to perform quite a lot of effort to finally locate the male gametes into the female reproductive system, but it makes the whole process of fertilization much safer because gametes never have to go outside, so the probability of them being destroyed by unfriendly environmental conditions is much lower. Well, and that's it. Ovaries, oviducts, uterus, endometrium, cervix, vagina. Much simpler than in case of male reproductive system. However, we are going to talk much more about what a female one does than about functions of a male reproductive system. Let's see what actually you have to know. The first of things that you have to understand is that becoming male or female decides basically at the moment of fertilization, but this is not fully true. Because of course you know that what determines the sex to the broad extent is proper set of chromosomes. So if you got two X chromosomes, then usually you turn female, while having one X and one Y in most cases makes you male. But this is not a matter of chromosomes as they are, but genes which are placed on them. And the very gene we are interested in is called SRY, and actually it is located on Y chromosome, so that's true. To have the proper gene, SRY, you need a proper chromosome, Y, to become male. All embryos start as females in terms of a set of hormones produced and organs and so on, and they develop as such. For some time, affected by female hormones produced by maternal organism, and they develop for some time, and then if there is Y chromosome present with SRY gene, then at some moment it will turn on, and activity of SRY gene will lead to extensive production of testosterone. Testosterone is what converts previously female embryo in a male one, and from this moment on it should develop as a male. So actually what determines the sex of the embryo, fetus, and future organism is not presence of testosterone, but its proper excess when compared to the level of estrogens, female hormones. If levels of testosterone are much higher, then the embryo develops into a male. If estrogens exceed testosterone, then the embryo will remain female. And in normal circumstances, presence of testosterone is determined by presence of SRY gene on the Y chromosome. But there are many situations in which something may be wrong with this determination, and it may lead to complications of this simple mechanism. For instance, part of Y chromosome can be translocated onto another chromosome together with SRY gene. In such a situation, the individual may not possess Y chromosome, but still it may possess SRY gene and develop into a male. Another situation is created by hormonal imbalance during pregnancy. For instance, if mother will take high doses of testosterone, as it may happen, you probably know, sometimes in sport, because testosterone may be used as sort of doping, then it may happen that the embryo, which actually should develop as female, will convert into male due to the presence of testosterone. But there is also the opposite possibility. For instance, if there is a real male embryo developing into a male, but mother is taking high doses of estrogens for therapeutic purposes or from other reasons, that it may happen that development into a male embryo will be somehow blocked, and it will still continue as female even if there are all necessary genes and hormones present, but they will be just hushed up by presence of excessive estrogens. So what I want you to understand from that is the basic sex determination is as that proper set of chromosomes X and Y is necessary to make the embryo male. It is due to the fact that there will be proper gene which determines high testosterone production at the proper moment, and if it happens, then originally female embryo converts into the male one and goes along this way. It should be like that. Testosterone may do different things. Before the birth, of course, it will control proper development of male gonads. So presence of the testosterone will take care of embryonic gonads, which are at the beginning sort of not decided. But due to the effect of testosterone, they will convert into testes and work as male gonads further. At the same moment, testosterone will also control proper development of all other reproductive organs. Then, for some time, there is no more interesting things to do for testosterone, and the next moment when it becomes dreadfully important is puberty. At this moment, testosterone levels will increase dreadfully, and it will trigger sperm cell production and also development of so-called secondary and tertiary sexual characteristics. Secondary sexual characteristics is proper development of sexual organs related to puberty. Tertiary characteristics are all these features which are not directly involved in reproduction but still are typical for the given sex, like voice mutation and the timbre of voice dropping, presence of facial hair more extensive than in females, muscle growth to bigger extent than in females, and also longer time of bone growth, what usually leads to the fact that males tend to be higher than females. And of course we know that all these features are not dependent only on the presence of testosterone, but also many other factors, and they are also dependent on the concentration of testosterone. So actually these are not things working in terms of all or none, but rather in the continuous way. Um, but basically, we may say that typical for the male sex, excessive levels of testosterone will at puberty drive the young male organism towards all these features more expressed than in average female. This is more or less what you have to know about testosterone action. So what it does before the individual is born, and what it does after the individual is born. And as you can see, this is not much about what testosterone can do. It is rather general, and that's enough because we are going to learn much more about what female hormones can do. This diagram is only to show you that actually this whole hormonal control of what happens in the male organism is much more complex, but you don't have to know about that. If it comes to female hormones, there will be, of course, quite a similar set of processes which will be controlled by female hormones, among which estrogens are the most important. So similarly to testosterone in males, estrogens in females will control proper development of female reproductive organs before the individual is born, and after the girl is born, during puberty, estrogens will determine secondary and tertiary sexual characteristics. And except of proper development of reproductive organs, it will be, for instance, breast development, and a bit different than in males, fat tissue location in organism, which is necessary for support of future pregnancy. So, to this extent, what estrogens do in females is similar to testosterone does in males. But still, we have to remember that actually this is not like in females we've got estrogens, and in males we've got testosterone, and they did separate things into separate organisms. Both estrogens and testosterone are present and produced by both male and female sex. But in typical male organism, levels of testosterone will exceed levels of estrogen, while in female organism, it's usually the opposite way around. So this is about ratio between hormones, not just their presence. Please do note that. Up to this moment, it was easy. Now, the dance hard part. Separately from all these things, female hormones have to control gamete production. Of course, it was the same in males, but in females it is much more complex, because while in males it is just to produce as many sperm cells as possible all the time, in females it has to be measured and precisely controlled, because if everything goes okay, then the consequence of female gamete production, it may be pregnancy. And pregnancy is a very long and hard and energy consuming process, and you cannot play lightly with that. So the whole menstrual cycle, as you maybe know, is to make sure that you don't go pregnant too frequently at wrong moment, and so on and so on. Now we are going to talk about how hormones help to control menstrual cycle and what actually it is. This diagram, as you maybe see, is quite similar to what was shown for males. We are not going to analyze this whole diagram. It is here only to remind you that what happens due to hormones in a male organism is quite similar to what happens due to hormones in a female organism. The difference, basically, is in the final outcome. But in females, one additional huge difference is overlapped with all what is beneath. Everything what is related to gamete production in females will be performed in a monthly cycle, mostly to make sure that only one oocyte is produced at the moment. There are 4 hormones we are going to discuss to understand how it works. 2 of them are produced by pituitary gland. If you remember what was said about hormonal control of the whole organism in general, you remember that pituitary gland tends to produce hormones which in turn control other endocrine glands. This is exactly how it is. Pituitary will produce 2 hormones which will affect ovaries. Then ovaries will produce their own set of hormones affecting the rest of the organism. Pituitary hormones are FSH, follicle stimulating hormone, and OH, luteinizing hormone. You can use abbreviations only, however, sometimes knowing the full name helps to remember what is done by each of them, because these names will be related to processes which are controlled by them. Then, if it comes to ovarian hormones, those which are produced by ovaries, then we have to know 2 of them, 2 types of them, because this is not one substance, the whole family of them: estrogens and progesterone. We are going in a moment to analyze how it works. The crucial thing to know is that their role is to coordinate what happens in ovaries and in uterus, because however both have a common interest, final production of the new organism, they are not directly connected, and if not communicating properly, then they not necessarily may do exactly proper things at proper moments to finally have a success in the form of fertilization and pregnancy. So to make it more likely to happen, they need proper communication in the form of hormones. And all these hormones work mostly due to the loops of so-called negative feedback. What you should remember from what we said about general hormone action in organisms. Many hormones work this way. If some parameter grows too far, then actions will be taken to bring it down. If the same parameter drops too far, actions will be taken to make it grow back to the desired level. So the final outcome is that in most cases the parameter will be more or less stable. Many such loops of negative feedback will be present also in the control of menstrual cycle. Now try to focus. It will be hard. If you need, you can analyze the same part of the presentation many times. I will provide, of course, the proper set of exercises to help you to work with that. But anyway, this is quite complex thing, but worthy of knowing. If you understand that, for girls, it will help to understand how your organism works. For men, it will help you to understand how female organism works, what can be useful at many moments. Let's go. Menstrual cycle are actually 2 separate cycles. Ovarian cycle, which is focused on gamete production, and uterine cycle, which is focused on what happens in uterus with special stress on changes of endometrium. You remember this innermost lining of uterus. So gamete production is rather obvious. You need it to mix with another gamete and produce zygote. If it comes to uterus, I will stress once more that the proper development of endometrium is necessary to later create a connection between developing fetus and the maternal organism. This connection is called placenta, łożysko, and is a core of the placental mammals' ways of reproducing. When the developing embryo is actually grown into the maternal organism, and due to that it is supplied with all necessary nutrients and other substances very, very efficiently. So ovaries will do their own work, and uterus will do its own work. And in order to make it coordinated, sets of hormones will travel to and fro to make sure that everybody knows its own work. Let's focus first on ovarian cycle. First, we are going to analyze the cycle. Hormones will be overlapped without later, because this is only how various things are signalized. But before you learn how it is communicated, you have to know what actually should happen. So, first, there is so-called follicular phase. This is rather easy. During this phase, the new egg cell, called oocyte, will be developing and growing in the ovary. And it grows till it is mature, ready. At the same time, estrogen is produced. Then there is the breaking moment, ovulation. If oocyte is mature, it will get out from the follicle and drop into the oviduct, and then it can wait for fertilization. But this is not the end of what is important in the ovary, because the follicle from which the oocyte was released will be converted into so-called corpus luteum, and it starts the luteal phase. Luteal phase is basically waiting for fertilization. During this phase, progesterone is produced. So for ovary, the work is very clear, divided in two stages separated with ovulation. The first phase is producing oocyte, then ovulation, releasing the oocyte, then waiting if the oocyte is fertilized. Very, very easy, right? You can see all these phases if you look at the ovary, because growing follicle becomes more and more visible. So at the very beginning, even if it is tiny, gradually it will become so big that you can see it under the ultrasound examination, especially if it is fully mature. This is a really large bubble within the ovary. What you can see on these diagrams here and on micrographs are subsequent stages of follicular phase. So the follicle growing with oocyte inside. Then ovulation. Ovulation is releasing the oocyte. This is a rather short moment, and afterwards, in the place where previously follicle was, corpus luteum will develop, which is like a ruffled, fluffy thing, and it is only for hormone production. So still, even if there is no longer the oocyte, you can recognize the phase of the ovarian cycle by the presence of corpus luteum in different size and shape. It lasts for some time. At the very end of this phase, if there was no fertilization, corpus luteum will convert its so-called corpus albicans, which is actually degradation, and then corpus albicans disappears and the new cycle may start. At the same time, uterus has its own work. First, usually, we start analyzing the cycle with menses, which is just menstruation. This is discarding the old layers of endometrium. What was once grown, now it is scrapped off, with some amounts of blood is excreted outside, and endometrium becomes thin once more. This is the very beginning. After menses, there comes so-called proliferative phase. At this moment, endometrium is rebuilt. What actually means excessive divisions of cells. This is why this phase is called proliferative phase. Proliferation is many divisions. So endometrium grows in thickness. Many new layers of cells are added. Then, after reaching some moment, in addition to constant producing new cells, endometrium will enter secretory phase when glands open in endometrium, making it ready for implantation of potential embryo. So only if the endometrium is in so-called secretory phase, it is really ready to communicate with incoming embryo and form placenta. And after secretory phase, if there is no embryo appearance, no attempt of implantation, then it will be the end of secretory phase and new menses arrive. This is what happens in uterus. Now, how to coordinate it and why? Basically, this is the diagram we are trying to understand. But of course not at once, gradually. So, when we are in follicular phase, it means that the follicle is growing in ovary, becoming bigger and bigger and more mature. At the same time, uterus doesn't have to curve. It may focus on its own work, which is discarding old endometrium and performing menses. Because when follicle is rather small at the very beginning, it has nothing to do with uterus and no need for uterus to understand what's going on in the follicle. But of course it will change. As long as follicle is growing, there is a stage on which uterus should realize that this is a good moment to end cleaning and focus on producing new endometrium. So the signal should be sent from the follicle, like our oocyte is growing, and at some moment it may be converted into a zygote. So please, the uterus, start preparing for that. Such a signal triggers proliferative phase in uterus, and uterus starts generating new layers of cells. At the same time, our follicle keeps growing. So if it grows large enough, there is ovulation, and then the signal should be sent to uterus. Dear uterus, now there is mature oocyte waiting for fertilization, which may occur at any time. Please, dear uterus, be aware of that and make yourself fully ready for arrival of the embryo if fertilization is successful. So, after ovulation, ovary goes into luteal phase, but the signal sent to our uterus will convert proliferative phase into secretory phase. At this moment, ovary keeps sending signals. Prepare your glands. Fertilization may occur at any moment now. Be ready. So uterus produces glands, being in secretory phase. Endometrium is very active and waits for embryo to appear, which may happen or not. If it doesn't, if there is no fertilization, after some time of waiting, corpus luteum converts into corpus albicans and degrades. And it is sort of signal sent to endometrium, there was no fertilization. Stop waiting, do your own work. At this moment, of course, secretory phase is over and menses start. So this is basically what should happen. If ovary is producing the oocyte, at some moment it will send signal to uterus to stop cleaning and make endometrium grow. If oocyte is already ovulated, then ovary sends a signal to endometrium to be ready, because at any moment fertilized oocyte may arrive as the embryo and try to implant. And if there was no fertilization, the signal from ovary will be sent to uterus that there was no success and you may stop waiting. Right? Do you see? There are three crucial things that should be signalized very clearly to the uterus from ovary. And of course, this signalization will be done with proper hormones. The first crucial signal for the whole cycle will come from pituitary at the proper moment. Pituitary starts sending a signal, start producing a new oocyte. The signal will be sent from pituitary to ovary. And the signal is FSH, follicular stimulation hormone. Do you get the abbreviation? Follicle stimulating hormone, because it stimulates growth of follicle. So follicle with oocyte inside starts growing. At the same time, when it grows, it should start sending signal to uterus that maybe this is a good idea to stop cleaning and start growing the new layers of endometrium. But at the very beginning, when the oocyte is very, very small, there is no point in doing that. But the bigger it gets, the more important it is for uterus to focus on preparing for the new cycle. This signal sent to uterus is estrogen. Estrogen concentration will be proportional to the size of follicle. So the bigger follicle, the farther into follicular phase, the higher will be level of estrogen. This high increasing levels of estrogen at some moment will prompt uterus to start proliferative phase. At the same time, growing concentrations of estrogen have another work to do. This is negative feedback, because the more estrogen is produced, the stronger is reciprocal signal sent back to pituitary. And the signal is, okay, we are working, don't worry, you don't have to prompt us more. This is the inhibitory signal, what is indicated by the tiny rhombus here. It means that growing concentration of estrogen will inhibit further production of FSH. It is produced, but not on increasing levels, because low levels are enough. So FSH is still being sent to the ovary. Ovary will focus on making follicle grow. Growing follicle produces estrogens, and this estrogen will be read by pituitary as a signal, everything is okay, you don't have to stimulate us anymore. And FSH levels remain relatively low. At the same time, estrogen levels are increasing and increasing, because our oocyte gets bigger and bigger as long as it grows. Concentration of estrogen grows, and at some point it reverses its action from being inhibitory to being activating. It's just dependent on the concentration of estrogen. If it becomes very, very high, instead of inhibiting pituitary, it starts stimulating it. Due to high levels of estrogen, pituitary will receive signal, follicle is actually very big, it is ready, we can ovulate. Pituitary, at this moment, should control of releasing oocyte and formation of corpus luteum. And it does it by sending sudden burst of LH, accompanied by burst of FSH as well, but LH is much more important. Due to burst of LH, we've got ovulation and conversion of follicle into corpus luteum, so the start of luteal phase. Both of these things happening due to high concentration of LH produced for a moment. Of course, if it happens in ovary. At the same time, the signal should be sent to uterus. There was ovulation, at any moment fertilization may occur, make ready, so enter a secretory phase. How is it done? Mostly by corpus luteum, because corpus luteum will start producing proper hormones. These hormones will be sent to the uterus as a signal of waiting for fertilization. As long as corpus luteum is present, it will keep sending signals to the uterus that glands should be working. These signals are two hormones, progesterone and estrogen. Estrogen is exactly the same hormone as during the first phase, but progesterone appears only in the luteal phase, and progesta means for pregnancy. So this is this very hormone related directly to ability to go pregnant, because it turns on uterine activity in terms of ability to produce placenta. As long as progesterone and estrogen are sent from corpus luteum, uterus will remain in secretory phase. But progesterone and estrogen will also have the reciprocal activity on pituitary, because their presence and their increasing concentrations will be received by pituitary as inhibitory signal. Dear pituitary, don't start the new cycle because we are still waiting for fertilization. So luteal phase is not only making uterus active and working for fertilization, it is also preventing pituitary from starting a new cycle. But of course we know that if there is no fertilization, then corpus luteum will turn into corpus albicans, which is no longer capable of producing progesterone and estrogen. It will be a signal to end everything. Progesterone and estrogen are no longer produced. Pituitary is no longer inhibited, while uterus is no longer prompted. So what are consequences? For uterus, this is a signal. No fertilization. Well, you can start new cycle, and uterus enters new menses. For pituitary, this is a signal. There was no fertilization. Start a new cycle, and our pituitary will provoke production of a new follicle. IB may want you also to analyze changes of individual hormones concentration during the whole cycle. So here you've got some diagrams. Skip the uppermost one because it is not important for you. Look in the middle graph. You can see what happens to FSH and LH. As we said, at the very beginning of the new cycle, FSH level increases because pituitary is sending signal to the ovary to start the growth of new follicle. But growing follicle reciprocally inhibits pituitary, so FSH levels are not increasing anymore and they stay relatively low. And later they even drop slightly because a follicle is really huge and it sends really large amounts of estrogens. And then, if it is mature and amounts of produced estrogens are excessive, then it turns the regulation upside down and prompts pituitary to start ovulation. So very large follicle will cause shot of both pituitary hormones LH and FSH. Due to that, ovulation occurs. Once it happened, LH and FSH levels will drop and they will remain close to zero the rest of the luteal phase. They will start increasing when presence of estrogen and progesterone will be much lower due to degrading of corpus luteum. If it comes to levels of estrogens, it is very easy to remember how it is. In the first phase, the bigger is follicle, the higher is level of estrogens, up to the moment right before the ovulation. Then level of estrogens drops slightly because there is no longer the follicle to produce them and corpus luteum is developing, but later estrogen levels will drop once more and will increase once more, and then it will drop till the end of the cycle. So basically you can compare the shape of the estrogen graph to the camel. The first hump grows till ovulation and then drops. The next hump will be in the middle of luteal phase and then drop. If it comes to progesterone, it's the easiest thing to memorize. Totally absent during the first phase because the only structure capable of producing progesterone is corpus luteum. Progesterone will appear together with corpus luteum and its concentration will gradually increase. Then it will start decreasing when the corpus luteum starts converting into corpus albicans. That's all. Easy, isn't it? By the way, changing levels of hormones will affect not only ovaries, uterus, and pituitary gland. It will also come into interplay with many other processes in the organism. Behavioral aspects, as you know, that when the levels of hormones drop, you become more, emotional. But there are also some physiological changes dependent on these hormone jumps and drops. For instance, the moment of ovulation is usually related also to the temperature increase. Body temperature will increase because it creates more favorable conditions for fertilization. So quite often you can detect the moment of ovulation if you measure your temperature regularly each day, because then you will detect that your basic pre-ovulatory temperature is some, but it increases about half of the degree after the ovulation. So if you know that your basic temperature is, let's say, 36.4, then after ovulation it will sometimes go close to 37. Then you know that it is after ovulation. If somebody tries to control fertility in natural ways, measuring body temperature is one of necessary steps to do that. It's not very good way of preventing pregnancy, but if you want to get pregnant, then it is absolutely necessary to know when you ovulate. Now look at this diagram, you know it already. Now try to understand it a bit. Pituitary gland produces FSH and LH. Both of these hormones will work in ovaries. FSH basically focuses on production of gametes, while LH basically focuses on preparation for pregnancy. This is the basic scheme of how control of menstrual cycle works. I assume it was not very hard. In the presentation, you will also find some further analysis of how levels of hormones change over the menstrual cycle, so use the presentation, the diagrams, the descriptions to absorb all this in the way which most suits you. The last thing is what you should know about in vitro fertilization, and it seems a bit stupid because you don't have to know how the real fertilization looks like, this biological one. So first we have to say some things about biological fertilization. This is the moment when oocyte is met by sperm cells, and from this moment on there will be a series of processes which finally should lead to the injection of genetic material of one sperm cell into the oocyte, connection of genetic material from the oocyte and sperm cell, and formation of a diploid nucleus of the first new cell of the new organism, the zygote. This is basically what fertilization is, and in normal circumstances it should happen in an oviduct. But in many cases it may not happen. If it is like for some reasons the couple cannot get pregnant, then it may be assisted by the range of medical procedures, one of them being in vitro fertilization. The first thing is that it is not always needed, even if a couple is infertile. Please do remember that infertility is always the couple's issue. So it may be like the same person may be fully capable of producing offspring with somebody else, but as long as is not able to produce offspring with this partner, we say that the couple is infertile. But of course, infertility causes may be at both sides or mostly on one of them. We are not going to analyze what are various reasons that may finally decide about performing in vitro, because IB is not interested in that. They only want you to know how knowledge about hormonal control helps to assist in fertility by in vitro fertilization. In this procedure, hormones will be used to subsequently stop female menstrual cycle in order to start producing many oocytes, then provoke overproduction of oocyte, then collection of oocytes, then collection of sperm cells, then making them meet in a dish. This is why we call it in vitro fertilization, because the environment for the meeting of oocyte and sperm cells is created artificially. Then the created zygotes will be incubated in order to see if they develop properly, and then the embryo will be implanted. Let's talk for a moment about each of these steps. The biggest problem, the biggest burden, the biggest work in vitro fertilization is at the female side, because if a couple decides for in vitro fertilization, a female organism should provide as many oocytes as possible. Normally, it produces only one, and the whole menstrual cycle is focused on controlling the whole process of producing one and one only oocyte by inhibiting the rest of follicles at the same time. Here, for in vitro, we want it different. Why? Because generally the whole procedure is burdening, invasive. Large doses of hormones are burdening for the organism. Generally it will make you sick. So if it has to be performed, it should be performed once, and due to that you should provide as many oocytes as possible, which they can be stored later by freezing, and it is no harm for them, but it prevents mother from suffering repeated cycles of hormonal dysregulation. So basically, if in vitro is being performed, the first focus is to try to provoke ovaries of a female to produce as many mature oocytes as possible at the same time. It is not a natural process, so it involves rather dreadful doses of hormones given at proper moments. First, proper set of hormones, which you don't have to know by the name, will be given to stop the normal cycle. Due to these hormones, no longer any follicle is developing in the ovary, right? Then the next step is giving very high doses of estrogens, what provokes so-called superovulation, development of many follicles at the same time. So the ovaries are bursting with oocytes, which, as you maybe remember, are really huge. And then when controlling physicians decide that oocytes are ready, HCG, which is pregnancy hormone, will be injected to loosen oocytes in their follicles and provoke ovulation. So at least 3 different abnormal hormonal procedures will be performed to get the abnormal biological consequence in terms of many follicles maturing at the same time and ovulating at the same time. But we don't want them to be ovulated. Right before ovulation, the egg collection is performed. At the same time, men should provide sperm cells. If it is impossible, because infertility is mostly on the male part, then cells can be taken from sperm bank. And now another thing which is quite often not known: sperm cells are not active before they go through the female reproductive tract. So they need this process of traveling across the cervix and then uterus in order to become ready for fertilization. And of course, if we perform in vitro, there is no such a travel across the uterus, so the sperm has to be processed before that. There are many different steps of this preparation, so-called capacitation, which makes them ready. Also concentration of sperm cells to make sure that many healthy sperm cells will be present, and so on and so on. And then, if we are sure that sperm cells are waiting ready, we collect egg cells. It is done by the wall of vagina with a special needle, and it is assisted by ultrasound examination to make sure that each swollen and mature follicle will be found and each egg cell will be picked up. Then, if we've got a collection of egg cells and collection of sperm cells, the basic procedure from this moment on is rather simple. We take a dish. We put each egg cell separately on the dish, mix each of them with some bunch of sperm cells, and leave them overnight to incubate. because fertilization is not a moment. It needs some time. Then, of course, we have to check if fertilization has occurred and if there are embryos developing. In case of some assisting problems, we may, for instance, perform genetic screening. In some countries, it is possible that if parents are burdened with some genetic disease, they can perform in vitro fertilization not because they are infertile, but because they want to choose the healthy embryo to be implanted, to avoid pregnancies resulting in children with sometimes very severe genetic diseases. Embryos are chosen for implantation due to their health. So, if they are properly developed and there is a high chance that they will develop correctly, then they may be chosen for implantation, and it is placing the embryo deeply in the uterus and then just waiting. Of course, afterwards the mother has to perform a pregnancy test to make sure that implantation has occurred, and the whole future pregnancy must be strongly monitored, because if there was a need for in vitro fertilization, that there is always assumption that this is high risk pregnancy. But in many cases, development of pregnancy from the moment it is established will be quite normal, and there is no longer any need to reach for hormonal support. But the very basic procedure of in vitro fertilization requires knowledge about how female hormones work in order to produce oocytes, and this is what is the main focus of IB in terms of what you should know about in vitro fertilization. Mostly, these hormonal procedures applied to mother in order to provoke hyperovulation. Here you've got just some more pictures. This is what you see here at this bottom left picture. This is how hyperovulating ovary looks like. So instead of one large follicle, the whole ovary will be converted into the bunch of mature follicles. This picture is created by ultrasound scan. So during the scan is performed, it is possible to insert a needle into each of these mature follicles and collect each individual oocyte, take it out. Oocytes don't have to be used instantly. If a couple does not decide to use all of them at the same moment, they can be frozen and wait patiently. There is also a possibility that fertilization will be performed instantly, but the embryos will be frozen and then implanted at the proper moments. Another set of pictures shows the procedure of in vitro fertilization in the dish. So, egg cell is present in each of these droplets. Sperms are added to each of these droplets. Then incubation gives sperm cells time for fertilization, and then if embryos are produced, they can be implanted in the uterus as deeply as it is possible. What you see at the bottom right picture is the oocyte right after in vitro fertilization. This is not a typical procedure because it didn't happen just in a dish. The so-called clamping needle was used, and these lines you can see across the oocyte is clamping needle holding the oocyte. So it means that probably the sperm cell was not left on its own to penetrate the oocyte, but it was injected directly into the oocyte. So of course the basic procedure we discussed is something what is the most frequently used, but there are also many other assisting procedures making fertilization even more likely to happen in case when the natural processes are for some reason insufficient. To summarize everything, we have to mention that what we know right now about reproduction in humans and other animals was not always known. And my favorite, William Harvey, the very same who performed such cruel experiments on animals to understand how their circulatory systems work, he tried to analyze also reproduction. For a very, very long time, it was thought that females actually do not participate in creation of the offspring. It was assumed that the essence of offspring comes from males and females only create environment for the development. It was called the seed and soil theory, formulated by Aristotle. It was also assumed that each sperm contains the tiny future baby, which can develop properly only if placed in the female organism. William Harvey decided to check if it is true. How it was done? He tried to did it on deer. Was maybe a bit less cruel than his experiments on dogs, because he worked on a game deer which would be shot anyway. What he tried to observe was if in the female deer embryo appears after the mating period. So these generally breed only in autumn. So Harvey went to gaming fields during mating season, and after he spotted the mating, they waited different periods and shoot a female, and then he looked in the uterus for any trace of developing embryo. And what did he find? Nothing. However, he tried really strongly. He shot many deer. Luckily, they were at least eaten later, so it was not such a waste of animals. He was not able to find anything interesting in the uterus of a female deer in shorter and longer periods after their mating. Any idea why? Oh well, poor man. He had no good microscope. So actually, there were embryos. Quite soon, maybe not like in minutes after mating, but in days after mating, embryos were already developing in the female deer he shot. But unfortunately, they were far too small to be detected without a good microscope, and he was not equipped with one. So, finally, it led him to the absolutely false assumption that whatever is the source of fetus created in the uterus, it does not come from neither male nor female. What, of course, was rather stupid, but quite logical in terms of what he saw. He saw deer mating, and then no new developing creature was found in the uterus. So he deduced that whatever is a cause of embryo creation is not directly the intercourse between male and female. He was misled. So I want you to know that, mostly to be aware that science is strongly limited by the tools it uses, and it always should be aware of that. The same person, William Harvey, was able to deduce presence of capillaries even if we couldn't saw him just from functions of arteries and veins. But unfortunately, there was not such a background of information in case of reproduction that could lead him to deduction what is the real process of fertilization without assistance of better tools than he had. Thank you a lot.